Now the very first chapter we are starting with is cell. Now this content is purely old and new NCERT. As per 2026 new class 9th NCERT, cell is the core chapter of class 9th NCERT. So yes, I have covered both old and new NCERT content. Okay, as updated as 2026, we also cover previous year questions related to cell chapter that came in both NDA and CDS examination. Okay. Okay, now we'll start the very first thing. Now, certain terminologies and discoveries. Okay, so the very first thing, the discoveries like Robert Hooke. What Robert Hooke discovered? So Robert Hooke discovered a cork cell, which is actually a dead cell. I'll just write it here, which is not written. Okay, cork cell is a dead cell. Remember. So Robert Hooke. No need to remember the year. Okay, it is not a history part, but yes, the name Robert Hooke. Okay, one more scientist is there whose name is Robert Brown. He actually discovered nucleus, cell nucleus. Remember, okay, cell nucleus. Then next was Schleiden. So Schleiden or Sheldon, he discovered, actually he gave cell theory. He both of them, Sheldon and Schwann, actually gave cell theory, in that they said that all plants are made up of cells, and Schwann said all animals are made of cells. That means the basic unit of life is a cell. So Sheldon talk about plants, and Schwann talked about animals specifically. Then next was Virchow. Virchow said that every cell every cell exists from, or you can say every cell is made up of from a pre-existing cell. Okay, so whatever cell we see right now, although it is very modified or in whatever means, there were a previous cell from which it developed. Okay, so the line given by that cells from pre-existing cells developed by cells from pre-existing cells are this line said by or given by Virchow. It is again a part of your cell theory. Okay. Next there was new term that is introduced in NCERTs, in later NCERT, that is thermophiles. Thermophiles. Now break down the root word. Thermo for heat. Thermo for heat. This fill matlab loving. So heat loving bacteria. That's it. Heat loving bacteria, very important. Okay, they can ask this terminology, I'm telling you. Thermophils, heat loving bacteria. Now calcium carbonate deposit from these unicellular organisms help to form the first protective cell membranes. Ultimately, they have these thermophils, that means heat loving bacteria, has a special coat of calcium carbonate. Remember, calcium carbonate coating. Okay, which obviously act as the protective layer. Okay, these are called thermophils. Then there was a terminology that is called totipotency. So totipotency, what does it mean? Just read the name. Maybe they can ask, but yeah, the terminology is more important. So Haberland. So Haberland in 1902, he proposed a term that is called totipotency. Now what is totipotency? The special ability of any living plant cell to develop into a complete plant. Okay, to develop into a complete plant, that is called totipotency. And this is the totipotency, this process, this terminology actually gave foundation to plant tissue culture. Now what is a tissue culture? Can anyone tell me? What you have heard this word no earlier? What is a tissue culture? Growing plants through plant tissues, right? In simple terms, without seed. Correct?
Yes, sir.
Got it? Yeah. Okay, good. So this is, in simple terms, this is called tissue culture. Okay, plant tissue culture. And totipotency is related to this one. It is the ability of a living plant cell. Okay, where cell grows into a plant. Okay, cell grows into a plant. It is called totipotency. Okay. Now in between class, if you have any doubt, any questions, you can tell me in between only. You can interrupt me. Okay. Now next, NCERTs now start mentioning about different type of microscopes. Very important. There are two types of microscopes: light microscope and electron microscope. They can ask you in examination which of the following is true statement regarding electron microscope. Okay, so light microscope use glass and lenses and light obviously bridges the micrometer gap. Ultimately, it can only view up to micrometer, but electron microscope is more advanced. It uses electron beams to see the tiny objects, and it can scale up to nanometers. Micrometer, how many? 10 to the power 10 to the power minus 6. Nanometer, 10 to the power minus 9. Got it? Clear, guys? Any doubt till now? Any doubt? Any questions? Clear to all of you? Yes or no? Fast, fast, fast. Yes, or
Yes,
no?
sir.
Okay.
Yes, sir.
Now there is a scale. Obviously, they can ask the question arranging them depending upon their sizes. Okay, proteins have quite a good size, or you can say they are very, you can say, smaller in size. Okay, millimeter, micro, and nano. Proteins are the smallest. So they can ask you, sorry, so they can ask you to categorize. So these are the largest. Okay, then in between and the smallest. Again, this is NCERT based data. So these are the updated new NCERTs class 9. So yes, they added these things, and there will be high chances they will create a paper on the basis of new NCERTs. So that's why we are covering this thing. Then again, certain formula is given: magnification equal to eyepiece into eyepiece power in multiplied by objective lens power, and estimated cell. How to determine the cell size? Diameter of a visible field in micrometer, and number of cells along the diameter. How many cell we can see, and ultimately the diameter of the visible field. This is how we estimate the size of a cell. Okay, like for example, how, I'll just give you a basic example. Like this is a wall here. Let us say, imagine this is the visible area. This is the wall. Let us say the area of this wall is 10,000 meters square. Okay, now this 10,000 meters square, now this wall is made up of bricks. This wall is made up of bricks. This wall is made up of bricks. Okay, now total there are, let us say, 1,000 bricks. Can anyone tell me the size of one brick? If the total area of this wall is 10,000 meters square and there are 1,000 bricks, can you tell me what is the area? What is the area covered by a single brick? What is the area covered by a single brick? Or what is the dimension, or what is the space occupied or area covered by a single brick? Or what is the size of a single brick? Can anyone tell me using this logic? If you understood, try. No one? It's very simple mathematics, sir. Very simple mathematics. See?
Maybe 10 meters, sir.
Yeah, exactly. If 10,000 meters square is the total area, number of bricks are 1,000. That means each brick is taking 10 meter square area or not? Simple. That's it. This is how they calculate the size of a cell. Now you got it. Okay. Got it or not?
Yes, sir.
Yes, sir.
Okay, nice. Next. Now next, there are two types of cells: prokaryotic and eukaryotic. Okay. Now what is the specialty? What is the difference between them? Okay. Now the basic difference between them is obviously the size: micro 1 to 10, and here 10 to 100. Comparatively, eukaryotic are bigger in size. Okay, usually unicellular. They can be uni and multicellular both. Okay, then primitive, these prokaryotic cells do not have a nucleus. Remember, they do not have a nucleus. Now, what does it mean? It is written here, na, nucleus is absent, but nucleus is there. Now, what is a nucleus? What is a nucleus in a cell? Like if I draw a cell here, and this is called the nucleus. What does it mean? Nucleus contains DNA. Okay, understood. We got it. Okay, that nucleus contains DNA, but this DNA, this DNA is covered by a membrane that is called nuclear membrane. So the region in which nuclear membrane is present, so this region, I'll just highlight it. So this region which is covered by a nuclear membrane is called nucleus. But in case of prokaryotic, there is no membrane. There is no membrane. So the genetic material, the DNA, the DNA is just floating inside the prokaryotic cell. That region in which DNA or genetic material is present is called nucleoid. So nucleoid is nothing, it's a region. It's a region where this DNA or genetic material is present. But if this region is covered by a membrane, then that region containing DNA is called a nucleus. Understood or not, guys? Tell me.
Yes,
Clear?
Yes,
sir.
sir.
Yes, sir.
Okay, good. Now again, the third, lacks membrane bound organelle. Lacks membrane bound organelle. That means whatever, first of all, prokaryotic cells do not contain organelle. That is the first thing. And second thing is that it contains only organelle that is called ribosomes. Ribosomes are only organelle present in prokaryotic. Present in prokaryotic. Other than ribosome, there is no organelle. Now, what is organelle? Organelle means, like for example, if there is a factory, if there is a huge factory, and in factory there are different departments. So those departments in cell are called organelle, and the factory is called cell, where different departments have their own role and functioning, but those different departments are not present in case of prokaryotic cell. Got it?
Yes, sir.
Clear?
Yes, sir.
Okay. Now, in case of eukaryotic, yes, membrane-bound organelles are present. What kind of organelle? Like endoplasmic reticulum, Golgi apparatus, endoplasmic reticulum, Golgi apparatus, mitochondria. These are the types of organelles that are present in eukaryotic. But prokaryotic, prokaryotic does not contain any kind of such organelle. Clear?
Yes, sir.
Okay, good. Now, as I said, it only contains ribosomes. Okay, ribosomes are the only organelle that is present inside the prokaryote. Example, bacteria. Example is bacteria. So
Sir.
bacteria is a prokaryotic cell. Yes, any doubt?
No, sir.
Okay, okay. Next, yes, we are done with this. Next. Now, okay. Now, I have one question for you guys. Now, prokaryotic says that lacks membrane-bound organelle, but I said that it contains ribosome. So, can anyone tell me, does ribosome contain any kind of membrane? Does membrane present in ribosome or not in prokaryotic?
Present.
Any logic? Just try with logic. Do they contain any kind of membrane? Yes or no? Obviously, no. Prokaryotic have lacks membrane bound organelle. So obviously, ribosome are also membraneless. They do not have membrane. Okay,
Yes, sir.
they do not have membrane. They are made up of proteins, and it is called ribosomal RNA. ribosomal ribosomal RNA. So these two are the constituents of ribosomes. They do not have membrane. Okay, next. Now next, viruses, viroids, and prions. Okay, what are these? Simple. These are the categorized as acellular. Why? Because they only contains genetic material. Genetic material is just floating. They do not have cell membrane or any outer covering. It is only the genetic material. But these three have certain slight variations in their structure. Very important. Again, NCERT, latest NCERT 2026 core concept. Okay. Now, not everything follows cell theory, obviously. Okay. Now, these are categorized acellular, no cells. Okay. And too small for a light microscope to see. Okay. Then viruses, composed of genetic material wrapped in a protein coat, only genetic material, either DNA or
Uh,
RNA. This is called genetic material. Next, viroids. Viroids are contains only genetic material but lacks the protein coat. They have protein coat. They have protein coat, but protein coat is not present here. Very important fact. I'm telling you, they will ask you which of the following is true for viruses, which of the following is not true for viroids. They will ask these questions in upcoming examination. I'm damn sure. Okay. Then in prions, misfolded proteins only, but genetic material is not there. I believe everyone can see the clear difference between viruses, viroids, and prions. Got it? Clear? Clear or not?
Yes, sir. Yes, sir.
yes,
Understood?
sir.
Okay, good. Good. Next. Now let's understand the plasma membrane. Okay. It is this plasma membrane that is ultimately present in every cell except these three. I'll just write it. Okay. No plasma or cell membrane. No plasma or cell membrane present. Okay. Now every cell contains plasma membrane. Every cell contains plasma membrane. Okay, the basic data, attracting obviously water in and water out. Okay, it is also called selectively permeable membrane because obviously selected things can pass through this membrane. Okay, now breakdown. What all things this plasma membrane contains? The very first part of plasma membrane that one need to know, that it is made up of phospho it is phospholipid, for phospholipid, or you can say bilayer. That means two layer. This one, I'll just highlight this two layer of phospholipid. Now one more trick I'll just tell you, that whenever you see this word lipid means fats. Lipid means fats. Okay, phospholipid. So bilayer of phospholipid. Okay, plasma membrane also contains proteins, proteins, or it is also called integral proteins. Why integral proteins? Because one part of plasma membrane joined by another part of plasma membrane, another part of plasma membrane by a integral protein. That's why they are called integral protein. That means joining. Integration means integration, integral joining. So one part of the plasma joined by a protein, that is called integral protein. Also, it has some traces of carbohydrates. Remember, these all things are present in plasma membrane. They can ask you four options. Which of the following is not present in plasma membrane? Listen these things carefully because we will practice questions in this class only. Okay, so make sure you listening these things very carefully. Now, also you have heard this word For fats, cholesterol, right? So sometimes these words are also used. Cholesterol related to again fats. Okay. Proteins can also be sometimes written as, sorry, not proteins, carbohydrates. Carbohydrates can also be sometimes written as sugars. Sugars means carbohydrates. Carbohydrates again, the basic unit of carbohydrates is glucose only. All things are made up of glucose. That is a C6H12O6, a group of glucose molecules called carbohydrates. That carbohydrates can be called sugar. A group of a combination of too much sugar is called starch, that is stored by plants. Now, talking about cell wall is present in plant cell. Okay, animal cell do not have cell wall. Remember this thing. Apart from plant cell, it is present in fungi and algae. And algae. Okay, in fungi it is made up of chitin, and in plant and algae both, it is made up of cellulose. It is made up of cellulose. And animal cell do not have this cell wall. Next, now there are two processes: diffusion and osmosis. First, I will talk about commonality. What is common between these two processes? The common thing between these two processes is that the material whatever passing through will go from high to low, will go from high to low. This is the commonality between diffusion and osmosis. But in diffusion we talk about gases, and in osmosis we talk about water. Diffusion does not need any kind of membrane. Okay, means it can happen without membrane or with membrane diffusion, but osmosis requires a membrane, that is selectively permeable membrane, or example plasma membrane or cell membrane. Now there are certain uh, examples. Okay, or there are different types of solutions. This one. Okay, now basic idea or basic movement is that, like, whenever there is a movement of water molecules. Okay, water molecules, obviously when we talk about water will go from higher concentration to low concentration. Just remember this thing. Now I will connect with you three types of solutions: isotonic, hypotonic, and hypertonic. Okay, now first let us understand hypotonic. Okay, then I will move to hyper, then we'll last see about isotonic. Now this hypo means, the root word hypo means concentration low concentration solution. Low concentration solution, remember this thing. Whenever I say low concentration solution, I'll just ask you one thing. You guys tell me. I'll take two beakers, one and two. Beaker A, beaker B, both have same amount of water. I added 2 tablespoons in A and 6 tablespoons in B. Can you tell me which of the following have low concentration, or which of the following is low concentration solution?
Yes, sir.
A1, right?
Yeah.
A, simple. It's very simple. Similarly, now in case of B, it will become more concentrated. So hyper means high concentration. Simple. And whenever one one trick is here, that whenever I say low concentration, that means water water is at high concentration. Water at high concentration. Whenever I say high concentrated solution, that means water will be water will
Oh,
be at low concentration. Now what does it mean? Now if water is high, remember, water is high outside the cell. Water is high outside the cell. Cell will swell. Okay, cell will swell. Water enters the cell, and if water enters the cell, the process will be swelling of swelling of the cell. Clear? Got it? And if
Yes, sir.
this swelling keep on happening, keep on going, it will not stop in animal cell. The animal cell will burst out, and that bursting out is called hemolysis. That bursting out is called hemolysis. Okay. Then next, and that obviously if water outside is at high concentration, that is called dilute solution. That is called dilute solution. That's why it is written then external solute is less than internal solute. Obviously internal solute of cell, internal solute of cell. Okay. So don't get confused with this. Okay. Just understand dilute environment. That's it. Dilute environment, water is high outside the cell. If water is high outside the cell, water will move from out to in. Water enters the cell, which will cause swelling, and this process is called hemolysis. This process is called hemolysis. Now coming to the hypertonic. Hypertonic, again, low water concentration outside. If water is outside low, then water will leave the cell and cell will shrink. If cell will shrink and ultimately keep on going, the process is called the process is called plasmolysis. The process is called plasmolysis. Clear? And it is a obviously concentrated environment. More high concentration, that means high hypertonic, more solute is added, shrinking of cell will happen. Okay. Clear? Then iso word means same. Iso word means same. No change in shape. No change in shape. Zero water movement. And no change in shape. Clear? Clear, guys?
Yes, sir.
Okay. Next. Next, talk about nucleus. Now, nucleus obviously contains a region that is called nucleus. Now, nucleus ribosome synthesis happens. The ribosomes we already know it is made up of protein and ribosomal RNA. Okay. Then next, now remember one thing, very important, very important. In normal conditions, there is a cell. This is the nucleus. When cell is not dividing, when cell is not dividing, very important difference between chromatin and nucleus. When cell is not dividing, whatever we see in microscope is like a round coiling of thread. It is coiling of thread, and that coiling of thread is called chromatin material. It is an entangled mass of thread like DNA present in non-dividing cell. But when cell start dividing, when then nucleus will also start dividing, and at that point of time, this chromatin material will start taking shape of chromosomes, and then it will divide. Chromosome are X-shaped rod structure during division. See, during division, and ultimately these each chromosome contain DNA, and one single part of DNA is called gene. Is called gene, and the structure is double helix structure. Clear, guys? Understood?
Yes, sir.
Understood? Any questions? Clear to all of you? Clear?
Sir,
Yes or no?
once
Yes.
explain chromatin, sir.
Sorry?
Chromatin.
Yeah, chromatin. See,
Yes.
chromatin material is nothing. It is a it is a coiled form. It is a coiled form of our chromosomes. Okay, it
Okay.
is coiled form of in simple terms, it is a coiling of DNA. This nucleus contains chromatin material. Remember this thing. This nucleus contain chromatin material when cell is not dividing in normal conditions. But when
Mm.
cell starts dividing, these chromatin material start taking shape of chromosomes. These chromosomes contain DNA. This DNA contains genes.
Okay, sir.
Got it?
Okay, sir.
Okay. Now, very important
Okay.
fact is written here that RBCs lack nucleus. Very important. RBCs lack nucleus. Okay, to make space for hemoglobin, but this limits lifespan of 120. So certain facts are also given here. Make sure you read it. Okay, clear? Clear,
Yes.
guys? Okay. Now let's understand a bit detailed about ribosomes. Okay, obviously found in prokaryotic mitochondria and chloroplasts specifically, and chloroplasts are present in plastids. Chloroplasts are present in plastids. Now there are types of ribosomes: 70S and 80S. 80S are present in eukaryotic cells. They are present in cytoplasm. That means stay in cytoplasm. Okay, and or RER. What is RER? Rough endoplasmic reticulum. Will cover. We have next slides regarding that. Now these 70S are made up of 50 plus 30. 70S. Now don't say sir 50 plus 30 is 80. Okay. Okay. So it is not the mathematics summation. Just see that like this only. Okay, 50 plus 30 become 70 and 60 plus 40 become 80. Clear? So what you need to learn, you need to know types. Obviously they will ask, is there any you can say 90S ribosome? No. Types are important. Where they are located is important. Okay, obviously small, dense, non-membranous, no membrane sites for protein synthesis. They make protein. Ribosomes make protein. Simple. Ribosomes make protein. Okay, can be free in cytoplasm. Okay, free floating cytoplasm for internal cell use, and bound to RER for transport. For transport. Now we'll go to the cell organelles. Endoplasmic reticulum. Now, endoplasmic reticulum, there are two types of endoplasmic reticulum: rough endoplasmic reticulum and smooth endoplasmic reticulum. Rough, why they are rough? Again, because of ribosomes. Ribosomes we already covered. What are ribosomes? Okay, ribosomes are protein making organelles. They
So,
are
where
non-members. Yes, yes, please.
where are ribosomes made
They are
or
pre-
manufactured?
they are present in nucleus. They are present in this endoplasmic reticulum. There is no such site for ribosomes to manufacture, but yes, when we talk about what ribosomes forms, they forms protein. But yes, there are certain specific kinds of ribosomes. They form by the combination of 50S and 30S type, that is called large subunit and small subunit. Again, where they are coming from, that is very detailed, no need to cover that. Clear?
Yes, sir.
Got it? Huh. Okay. So what we need to remember, that ribosomes are protein synthesis. They do the protein synthesis. They manufacture protein. Okay, they secrete protein. Okay, what they are made up of? They are also made up of protein and you can say this ribosomal RNA. Okay, where they are located and what are their types? That would be enough. Now next, their tiny protein synthesis happens. Now two types: rough. Why they are rough? Rough because of ribosomes. Smooth. There is no ribosome obviously, and they store fats. Fats are also called lipids. Stores fats and hormones. So two types of endoplasmic reticulum. Okay, rough endoplasmic reticulum manufacture protein. Smooth manufacture fats, lipids. Okay, now it will not store. As soon as manufacturing is done, it will transport to the Golgi apparatus. Now the Golgi apparatus do the modification, the sorting, the packaging into its vesicles. These are called vesicles, and these vesicles are called cisterns. These vesicles are called cisterns or cisternae. Cisternae. So the modification, the packaging, the storage of these protein and fats are done by Golgi apparatus. Okay, this is how they are linked with it. Clear? Now, who discovered? Obviously, Camillo Golgi. where he see the network in a barn owl nerve cell. He see this thing first time in barn owl nerve cell. Remember, it is again mentioned in NCERTs now. Earlier it was not there. So it is written as system A. Okay. Next. Now, mitochondria. Obviously, everyone knows powerhouse of the cell. Basic things are there, but this mitochondria contains inner two membranes. Okay, double membranes, double membrane, double membrane. Mitochondria contain double membrane. Apart from mitochondria, is there any organelle? Yes, nucleus also contains double membrane. Nucleus also contains double membrane. So here there is outer layer, okay, simple layer, and other is deeply folded, and this inner membrane, which is deeply folded, actually breaks down glucose into energy. Okay, this deeply folded membrane actually provides the surface area for the reaction. For in simple terms, ATP generating ATP generating reactions. It provide surface for ATP generating reactions. Now, there is a word that is called endosymbiosis. Okay, endosymbiosis. Both have, that means plastids and mitochondria. They both contains own DNA and ribosome. That means they are self-reliable. Okay, mitochondria can manufacture its own protein because it contains ribosomes. Because it contains ribosomes. Because it contains ribosomes. Simple. Then for plastids, three types of plastic: chloroplasts, we already covered. Ribosomes are part of it. Okay. Then chromoplast, colored, attract pollinators like yellow color, orange. Leuco is colorless. Store starch, oils and proteins. And chloroplast, obviously chlorophyll in the stromas. Stromas are a part of that region where chlorophyll is there. Okay, what kind of material or metal present in chlorophyll? It is magnesium. So these are the facts regarding chloroplast. Okay, now next there is another organism that is called lysosomes. They have cleaning system. They have the cleaning system. That means they contains digestive enzymes. They contains digestive enzymes. Now, where these digestive enzymes are coming from? These digestive enzymes are manufactured by RER, rough endoplasmic reticulum. Manufacture these digestive enzymes. Remember this thing. And interestingly, the human sperm use lysosomal enzymes at their head, because during you can say fusion of sperm and egg, the head of sperm burst out. The head of sperm bursts out, and this bursting is done by this lysosomal or lysosomes digestive enzymes. Very important. New NCERT added this fact. That's why it is mentioned here. Then vacuoles are the storage areas. So water, minerals. Okay, cell sap. That means waste material. Sorry, that means waste material. Okay, in plants it is large in size, occupy almost, you can say, 50 to 90% of space or volume. Okay, wilted plants, remember, wilted plants lack vacuole water. That means those leaves, or you can say plants, are fall off. They are not living. Okay, they usually lack this vacuole. Then there is a term that is called cytoskeleton. Now, what it is? It is present in eukaryotic only. It is a network of fine fibers. Fine fibers. What is the role of it? Structural support, shape maintenance, and internal transport. Visible only via electron microscope. These are again new NCERT introduced this term to class 9 students. Okay, just keep focusing on these words. We'll have a PYQ, I'm telling you. Okay, then you will realize like how much relevant we have covered. Okay, now next is your uh, types of, you can say, the divisions. First is mitosis. First is mitosis. Okay, remember mito for T for two, T for two. That means one cell divided into two daughter cells. Meiosis, obviously, one cell in first stage goes into two. Then there is second stage which leads to four daughter cells. One division, two division. Okay, two identical cells. Yes, two identical cells. Four distinct gametes. That means chromosomes. That means chromosomes become half. What does it mean? Here 46 chromosomes are there. That means 23 pair. Okay, out of which one are sex chromosomes. Okay, male have X and Y, female have X and X. Okay, so one pair goes to sex chromosome. Total 23 pair are there. There is, so if there is 23 pair, that means 46 in number. Then in mitosis, this number remain intact because it is exact copy and identical cells are there. But in case of meiosis, we will get 23, 23, 23, and 23. Half chromosomes will be there. Chromosomes become half. Why? Because they are present in sperm and egg. They are present in sperm and egg. So in fusion, during the fusion, 23 chromosomes brought by sperm and 23 brought by egg. Then only it will become 46. That means 23 in pair. Remember this thing. Very important. Then again, chromosomes maintained and it is halved. Okay, whatever I have explained, it is written here. Now there is cancer cells. Now very important, like how this cancer cells spreads, because this knowledge is also added, or this factor is also added in NCRTs. Now normal cells can stop dividing when they touch neighbor cells. Remember, this is the natural trend. This is natural pattern or behavior of a cell, that whenever they touch the neighboring cells, they will stop dividing. But in case of cancer, the cell lose this control and dividing uncontrollably to form tumor. The multiplication division of cells is so fast and so much that it keep on happening. It is uncontrollable, and that is called cancer. And that cancer causes, the cause of this cancer is tumors. The tumor is formed. Uncontrolled division of cells. Okay, important. Then there is programmed cell death, PCD. Organized cell suicide, vital for development. Eliminating tissue to form individual fingers. That means again, there should be a limited type of, limited size and division that should be controlled. If it is uncontrolled, then again it will become cancer. So body have this kind of mechanism that is called programmed cell death. This terminology is also added in NCERT latest. Very, very, very important. So it is called organized cell suicide. It is important for growth. Clear, guys? Any doubt? Any doubt? We are done with the chapter. Any doubt in any
Sir,
part? I'll just repeat it. Yes.
is every cell have their own chromosome?
Yes, definitely. Because see, chromosomes are present in chromatin material, and chromatin material is integral part of nucleus. So nucleus is there in every cell. So ultimately, chromosomes are also there. Got it?
Yes, sir.
Okay. Anyone else? Any other doubt? Any question? Any question? No? Clear? All of you? Clear, guys?
Yes, sir.
Okay. Okay. I'll just share. Now we'll do certain previous year question. You also do. First, you guys do, then I will help you. Okay. Now, these are NDA and CTS mix. Okay. Question number one for you. Try. Try.
B,
You
sir.
have to give the answer, guys.
B,
That
sir.
was the answer. Yeah. Which one?
B, sir.
B.
Yes.
So increasing of complexity, obviously organism is most complex. So these two options eliminated. Okay. Then C. Just we have covered that protein are the smallest ones. Protein are the smallest existing things, right? Right or wrong?
Yes, sir.
Yes, sir.
Obviously. Then if you talk about now difference between organ and tissue. Now remember one thing. Protein is there. Okay. Protein is there. Protein contained in cells. A group of cells. A group of cells form tissue. A group form tissue. A group of tissues. A group of tissues will
Oh,
form organ, and obviously organ contained in organisms. Now give me the right answer.
DT.
Got it? Clear? Understood? So
Yes,
protein to
sir.
cells, cells to tissue, tissue to organ to organism. That's why just we have covered no. See. Now it is your part to revise. I'm telling you. You have to revise it. See this one. Smallest, right? Proteins. Then you will see cells. So protein to cells. Then cells with tissues, tissues and organs to organism. Okay. Next. Now read it and I'll tell you how to solve. Now you guys try because you need to make some mistakes. When you start making mistakes, then only you will learn. Try and give me the answer. Read and solve. Okay. Anyone? Now even if you don't know some things, it's totally fine. You can still solve this. See, DNA replication takes place when chromatin is open. Chromatin is open. Okay, let us say it may be confusing to you. Don't worry. Chromatin organizes itself into rod-shaped chromosome before cell division. No, no. So two is wrong. Eliminated. See the way I'm solving the questions. I'm telling you. Observe me very carefully because this is the same method I used in my examination, and you guys will also do the same. The elimination technique. Okay, eliminate which are wrong. So, or chromatin organizes itself into rod-shaped chromosome. No, before cell division. No, after cell division. After cell division, because then only that chromatin material will take shape of a chromosome during division or after division. So this is wrong. To eliminate now it is one, either one or three. Now both prokaryotic and eukaryotic have the same process of cell division. Same process or DNA replication takes place when chromatin is open. Now try. What do you think? What do you understand? Try. Don't worry about mistakes. I'm telling you. But this is how
Maybe
you will
three is
learn.
right, sir.
Huh?
Maybe three is right.
Okay. What about others?
One, sir.
First
One.
one is correct.
Obviously,
One is
prokaryotic have different. They do not have same method of cell division. They divide
yes.
differently. Okay. So it is wrong. One is right. Don't worry. Now you learn. Simple. This is how learning happens. But first you need to make certain mistakes. Then only you will learn. Okay. Whereas how it is not same. For eukaryotic, we have mitosis and meiosis.
Yes, sir.
But for prokaryotic, there is no mitosis and meiosis. We have fissions. We have fissions like binary fission because it is asexual, or you can unicellular by binary fission, or if it is goes with more than two, so it become multiple fission. So binary fission is usually the method. But in eukaryotic, we have mitosis and meiosis. So it is not same. Next, read it and eliminate. Very simple. Very simple. If you remember, if you listen to me during the class very carefully, you can do it by elimination only. Try. Yes. Fast, fast, fast. I'm waiting.
D, I think, sir.
Okay. Now see, let's do the elimination. They are composed of phospholipid only. Now this only word is dangerous here. They are contain protein only. Eliminated. They are contain phosphorylated protein only. No. What about carbohydrates? What about cholesterol? So yes, option D. Simple. Carbohydrate is not mentioned here. So yes, cholesterol is there. So whatever data it is given, it is correct. But this only word is dangerous here. Are you getting me?
Yes, sir.
Okay.
Yes,
Next.
sir.
Next. Now try.
Consider a function regarding cell law, bacterial cell law, use plant cells. Sir, 1 and 2.
Okay, one and two, good. What about others? Fast. I want answers. Leave right and wrong. Don't bother about that.
1 and 3.
One and three, okay. Different answers, good. What about others? Fast, fast, fast. Come up, come up.
Option
One
C,
and
sir.
three.
Okay, let's see. Now, this I didn't mention. This I didn't told that bacterial cell made up of peptidoglycan. Just write it. Okay, that's the fact we'll cover. But you know that fungal cell wall is not made of cellulose. It is made up of chitin. Even if you don't know, at least you should know that it is not made up of cellulose. Obviously. So two eliminated. Gone. Gone. One and three done. Even then, this is the beauty. If you need to be a bit smart. Reading, revision, practice is one part that you will do honestly, consistently, wholeheartedly. But on the other side, you need to be a bit smart regarding solving of questions. And yes, three is also right. Like cell wall, obviously, and has extracellular made up of sugar and protein. What is this sugar and protein? Sugar is carbohydrates, and they are talking about plasma membrane. Isn't it plasma membrane? Yes or no?
Yes.
That's it. Simple. It is a language that troubles. But again, you need to be smart. So here, not only the knowledge part, but we will learn how to become smart simultaneously. Because that is required in examination. Okay, now this one. 69. 69.
Plasma.
Which one?
D. Plasma
D.
membrane.
D. Plasma membrane.
Okay. Okay, I'm getting one, two, and other. How many? Two answers on plasma. What about others? Give your answers. Give your answers. Fast. I'm
D,
waiting.
sir. D, sir.
D. Okay. One more. What about others?
Last one.
Okay. That means if one didn't use the brain, others also forgot it. So it is a nucleus. Okay. Let's see. See, I just told you that, see, nucleus absent. Now you remember. Now, why I said these things are important. Nucleus absent. Clearly written. So if nucleus is not there, what region is called? That region is called nucleoid. Okay. So yes, and
Yes.
one more thing, I also
Uh,
told one thing, that cell membrane is present in every cell. Okay, cell membrane is present in every cell. Remember this thing, except viruses. Remember?
yes.
See, I have written that no cell membrane, plasma membrane, or every cell contains cell membrane. That's why listening and reading both are very important. So for your reference, you must go through recording. Whenever you forgot, whenever you want to revise, must watch the recordings. So it is nucleus. Next, 70. Simple.
Mm, Implant. So, from the content.
Fast, fast, fast. Do it fast. Endoplasmic reticulum, obviously we didn't study. Golgi apparatus
Please.
we didn't study. Vacuoles we didn't study. Obviously C. C. Next. Try. Read this question carefully. No need to hurry. Okay? You are not going anywhere. I'm also not going anywhere. Just read carefully and then solve.
Not.
Cell wall.
B, sir.
Not which one? B. Not always present in a living cell. It is cell wall. Example, animal cell. Animal cell is living cell, but cell wall is not present in it. So not always. This is the keyword that you need to catch here. Now you might be wondering, sir, you told cell membrane is not present in viruses. Array, viruses are not living cells. They are not even cells. That's why they are called. Got it?
Yes.
That's why see how precisely you need to catch those words, and it will not come with one or two reading, successive reading. Okay, continuous reading
Yes,
and
sir.
revision and practice, because you're not able to recall every terminology in one go. Practice is there. Okay, but don't worry, we'll do a lot of practice. Now this one, try.
Yes.
B, sir.
Yes.
B.
Hypertonic solution. High concentration, that means low concentration water outside the cell. If outside the cell water concentration is low, that means water will flow from the cell to out. Cell will plasmolyze. Option B.
Yes.
Clear? It will
Yes.
not swell. You do not know about the turgor pressure means internal pressure. Obviously, no. Cell size unaffected, no. Got it? It will
Yes.
plasmolized. Next, try.
Cells of Yes, sir.
It is isotonic. It is isotonic. No change.
Okay.
Reading, read it carefully again. Practice. This is how you will practice it. Okay. Next. Okay. Now this is important. I'll just tell you. Just mark this. That it is present in. No. Wait. It is present in this. Not endoplasmic reticulum. Endoplasmic reticulum, I think, does not contain. Yes. So option D. We just write it. Important. Yeah. Endoplasmic reticulum is also part of it. Just write option C. Option C. Sorry. Option C is also because endoplasmic reticulum also contain RNA. Example. Why RNA? Because it contains ribosomes, and ribosomes are made up of ribosomal RNA. Okay, that's right. Option C. So write it. Or you, okay, that RNA is present in nuclei, cytoplasm. Why cytoplasm again? Ribosomes are present there. Ribosomal RNA, ribosomal RNA, mitochondria, chloroplast, obviously, because wherever DNA is there, RNA will be there. Common sense. Okay, clear? Clear, guys?
Yes, sir.
Okay, next. Try. Expelling excess water. Golgi storage, endoplasmic reticulum transport, lysosomes, suicide bags of the cell. Okay, lysosomes are also called suicide bags of the cell. Okay, obviously vacuole. Vacuole is the right answer. Clear? Got it?
Yes.
Yes, sir.
Okay. Next, try this. Another good question. Again, logic. ATP synthesizing, ATP generating chemical reactions. Remember, ATP is generating chemical reactions. ATP generating chemical reactions. Inner membrane. Two membranes were there. So inner membrane. See, if you don't remember, I'll just show you. Okay, I'll send this PDF. Make sure you read it. Yeah, this one. Got it? Clear?
Yes.
Okay,
Sir.
next.
Human body is used in tools for some.
Now I'll just give you a hint. It is a hypertonic. It is a hypertonic solution. Look. Now solve. 2% detergent solution is hypertonic for blood cell.
It swells.
B, sir.
Huh? In hypertonic,
It swells.
cell swells or shrink?
Swells.
It shrinks. See? Again, revision. This is the important part. Revision. See, hypertonic. What is happening? Shrinking. Got it?
Yes, sir.
Okay. Next. Lipid synthesis. We covered RER, SER. Remember?
Yes, sir.
Smooth endoplasmic reticulum. Okay. Next. Okay, now next 72. Try. Oil stores starch, oil, and protein granules. Chromo for color. Xantho, we don't
Leucoplast.
know, leave it. Chloro for photosynthesis. Leucoplast. Okay, now next 73. Try 73.
C.
Not correct. They have asked not correct. So, occupy large space, yes. Vacuoles provide turgidity and rigidity, yes. Vacuoles are absent in animal cell. No, they are present in animal cell also. And amoeba vacuole role in nutrition, yes. How? We'll cover in upcoming chapters. Okay, so in amoeba, vacuoles are called food vacuoles. Okay, the vacuoles in amoeba are called food vacuoles. So yes, their role in nutrition. So the vacuoles are present absent in animal cells. No, they occupy 0 to 5% space. So they are not absent. Not correct. हो जाएगा. Next. Now try this one. This is CDS previous question. Again, elimination, guys. Read it carefully. Eliminate. Bilayer of proteins. No. Bilayer of neutral lipid. No. Bilayer of neutral lipid. No.
Yes, sir.
That's it. Finish. Catch the keywords. Okay. Next. Try. So in plant cell is eukaryotic cell. So plant cell is eukaryotic.
Yes.
Okay. So if it is eukaryotic, so nucleus does not need a membrane. No. Nucleus and
B
chloroplast
part.
are surrounded by separately their own membrane. Yes. Surrounded by membrane, but chloroplast do not. No. Nucleus are not. No. Eukaryotic, obviously, there are membrane bound cell organelles. Remember, membrane bound cell organelles.
Yes, sir.
Yep. Okay. Next. Dietary enzymes are present in fast, easy,
Vacuum.
Lysosome.
Biosomes. Obviously. Next.
Which one?
Read it
Yes.
carefully. Very easy.
C part. Cell will swell up.
Now, they said that animal cell is surrounded by a low concentration water. Cell is surrounded by low concentration water. That means cell will lose water.
Hmm.
Obviously, water will move from high to low, inside high,
Hypertonic.
outside low. Got it?
Yes, sir.
Okay, next. NDA, previous year.
Nucleoid.
Huh? Nucleoid, remember? The region, undefined nuclear region.
Yes, sir.
Okay. Nucleus is present in nucleus. See, I'll just show you. Everything is here. See, Got it? Clear, guys? Okay, next. It's very simple. Do it. Yes, fast.
B.
Okay. D or B?
B, sir. B.
B. Animal cells have only cell membrane but not cell wall, but cell level. True. Done. Okay. Next. Now this one again very simple. Eliminate. Now you know a lot of things.
Second and fourth.
Second, sir.
Okay. That means this one, option B. Option B. So all
Sir,
cells possess a cell wall? No. All cells have well organized nucleus? No. There is a region undefined that is called nucleoid. All cells have their linear DNA molecule present in them. Again, now see, very important. All cells have their linear DNA molecules. One option, another is two. All cells have ribosomes present inside. Yes, ribosomes are present. Ribosomes are present. That means two is right. Two is right, but again, what we eliminated one and three. One and three both go. Now two contains A, B, and C. Oh, sorry, one and three are also there. Now four.
Four.
Now linear DNA. What is this linear DNA? Can anyone like what do you understand by this by default? Like what is all cells have linear DNA molecules? Hmm?
linear DNA means one helix means RNA.
Yes, you can say that. Okay, it is present. Ultimately, it is nothing new. It is the same DNA. Ultimately, the DNA double helix structure present in the cell. Similar thing. Okay, so both 2 and 4 are right. Okay, don't get confused with this. Linear DNA molecules are the same thing as DNA. They are present in eukaryotic cells. Okay, eukaryotic cells contain your animals, plants, fungi, algae, all of these. Okay, it is present in eukaryotic. Same thing, nothing different. Next. Okay, now this is latest. No, this question I think linear. This is for current NDA 2026 question. Okay, this is latest NDA 2026 and this is your CDS question. Okay. Now before doing this question, I'll just explain you one thing here. Very important. Now, whenever we talked about viruses here, when we talked about viruses,
Wait.
this one. Now, viruses, uh, they usually considered dead whenever they are outside outside the cell or host cell or host body. Okay, and whenever it enters in any cell, it hijacks the nucleus. It hijacks the nucleus and uses cellular cellular machinery and uses cellular machinery. Okay, but when we talk about antibiotics, now how these antibiotics work? Antibiotics work upon bacteria. Remember, work work on bacteria, not not on viruses. Why? Because these antibiotics, you can say, break down break down cell wall manufacturing break down cell wall manufacturing system or machinery. So viruses do not contain any kind of cell wall. So antibiotics do not work on viruses. They work upon bacteria. So they attack the cell walls or cell wall making mechanism. So now you can answer this. Try. Now try. Because it targets
Bacteria.
cell wall. Got it?
Yes.
Okay. Next.
Yes. Total cytoplasm.
It is serious question. Latest 2026.
Nucleoid.
Nuclear.
Again, nucleoid. Same thing.
B.
Prokaryotic. Central cytoplasmic region. Okay. Nucleic
Nuclear.
acids containing nucleic acids are ultimately DNA, genetic material. Now you getting the terminologies. Getting it or not?
Yes.
Okay, good. Next. Okay. This I will tell you. This is a new question. I want to cover it. First of all, cover this. I'll cover that part. First, do this one. It's easy. Yes. Answers. Vacuoles. Okay, leave it. Mitochondria.
B.
Cellular respiration.
Respiration.
Respiration means generating ATP. Okay, got it. Remember this thing, right? Now, smooth endoplasmic detoxification in liver
Okay.
cells. Okay, because they forms lipids, lipids, and detoxification in liver cells. Okay, detoxification is also one of the function. Then cytoskeleton.
Yes.
Cytoskeleton.
Storage.
Maintenance of cell shape.
Structural. Structural. Okay, so we already made A4. So A4 and C2, A4, C2, B1, D3. Got it?
Yes.
Maintenance cell shape. How? How again? See, Cyto. They already asked actually. See, this is CTS 126 paper. Maybe in next they will ask in your. Yeah, this one. Structural support, shape maintenance.
Hmm.
Got it? Clear, guys?
Yes, sir.
Okay. Maintenance of shape cell. So option D. Next, try this. NDA. NDA question.
Mitochondria and chloroplast.
Simple. Repeated question. See, this similar type of question we did it or not? That was back in 2022 or 2023, and in 2026 in NDA they again repeated it. Questions do repeat, especially in science. This
Mitochondria.
ribosomes. Simple. Again, this one.
Due to the presence of ribosome in its
Ribosome
surface.
on its surface. Simple. Ribosomes. Simple. Okay. So one question is left. Now, not a function of pigment carotenoid. You just write it. Now, carotenoid are responsible for yellow and orange hues in autumnal leaves. Yes, true. Protecting chlorophyll from photooxidation. Yes, especially chlorophyll A. There are two types of chlorophyll: chlorophyll A and chlorophyll B. Chlorophyll A actually do photosynthesis. Photosynthesis. Then photoreceptive role in our eyes. Yes, it is also present in humans. The reaction center in pigment system. No. So this is not the function. Okay. So correct answer will be D. Just write these in your notes because it is in form of questions. Got it? So chlorophyll A actually do photosynthesis. Remember this thing, guys. Okay, clear?
Yes, sir.
Okay. And okay, we can also write some more facts. Like there is chlorophyll C, chlorophyll C, chlorophyll C found in marine algae, or brown algae. Okay. Or chlorophyll D is present in cyanobacteria. Cyanobacteria, which is also called blue green algae. Okay. And green again, it contains green, so it also contains chlorophyll A. Clear? Here, guys,
Yes,
it is
yes, sir.
present in. It is also present. You can also write red algae. Red algae. So algae is red because of chlorophyll D. Because obviously you can say it absorbs most of red light. Okay. Clear? Got it? So we are done with